Photosynthesis is one of the most vital biochemical pathways since almost all the living organisms depend on it for nutrition directly or indirectly. Photosynthesis is the process by which several living organisms utilize the solar energy for growth and metabolism. By definition, photosynthesis is described as the process of converting light energy into chemical energy by living organisms. The process of photosynthesis uses raw materials like carbon dioxide and water, and solar energy and produces oxygen and carbohydrates. Higher plants, phytoplankton, algae, as well as some bacteria carry out the process of photosynthesis. Photosynthesis occurs in two main stages, the first stage being the light reactions and the second stage being the Kelvin-Benson cycle. In short, the light reactions capture the light energy and utilize it to make high-energy molecules, which in turn are used by the Kelvin-Benson cycle to capture carbon dioxide and make the precursors of carbohydrates. This phase of photosynthesis is the initial stage, which converts solar energy into potential energy. This step requires ample sunlight for the process and hence it is termed as the light-dependent phase of photosynthesis. The light-dependent reaction converts He, P and N, He, D, P plus into the energy carriers He, T, P and N, He, D, P, H and produces oxygen. During this conversion, the chlorophyll's electron flow can either be cyclic or non-cyclic. The cyclic electron flow, also called the cyclic phosphorylation, involves electron transfer chain that starts from a pigment complex called photosystem I in the chlorophyll. It passes from the primary acceptor to ferredoxin. From there it is transferred to a complex of two cytochromes and then to plastoquinone, after which it returns to chlorophyll. This electron transport chain produces a proton motive force, PMF, which leads to the pumping of H+ ions across the membrane and results in a concentration gradient that is used to power ATP synthesis during chemiosmosis. The cyclic pathway is seen in bacterial photosynthesis. The non-cyclic photophosphorylation is a process that involves two different chlorophyll photosystems. This process also occurs in two steps. In the first step, a single water molecule is broken down into two hydrogen ions, half a molecule of oxygen and two electrons by a process called photolysis, which literally means light splitting. The two electrons obtained from this reaction are kept in photosystem II, while the hydrogen ions and half oxygen molecule is kept for further use. After this, a photon is absorbed by the photosystem II, which results in transfer of two electrons to the acceptor molecule. The resulting deficit of electrons is replenished by accepting electrons from another molecule of water. The electrons then transfer from the primary acceptor to plastoquinone. Then the electrons are transferred to plastocymine, resulting in the production of the proton motive force. The light-independent reactions, or the reactions of the Kelvin-Benson cycle of photosynthesis, occur in the stroma. These reactions do not require the presence of light and hence the name light-independent reactions. In this cycle the end products of the light-dependent reaction are used. The light-independent Kelvin-Benson cycle reactions have three phases, which are carbon fixation, reduction reactions and ribulose 1.5-biphosphate regeneration. The immediate product of the Kelvin-Benson cycle is water and glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate molecules that have exit in the cycle are then used to make larger carbohydrates.